Today I want to ask a really simple question, or what seems like a very simple question, which is, what are our bodies? We've got these things, what are they? And that's probably the best initial response. We all know what our bodies are. Why are we talking about this? And in particular, we know this about our bodies. One, each of our bodies are very personal. So mine is mine and yours is yours. And we may get close to each other, but we don't end up with the same body. We just have one of them. We don't have two or three of them. At least at any one time, we just have one of them. They're persistent in some sense that's very difficult to define. So mine has persisted for a long time. Some of yours have persisted longer or shorter. And philosophers have argued for millennia about in what sense our bodies persist, since, obviously the components of our bodies change a lot from one time to another, and all the molecules in our bodies change. Cells come and go, et cetera, et cetera. So what we mean by persistent isn't too clear, because things are always changing. And sometimes they change in pleasant ways, and sometimes they change very quickly in very unpleasant ways. And philosophically, we've always asked questions about our bodies. So here are just some options from the history of Western philosophy. We have materialism, where we are our bodies, or dualism, where we inhabit our bodies, or a kind of straightforward reincarnationism, where we inhabit bodies in series. And of course, there's idealism, where we just imagine our bodies. And people hold these theories more or less seriously. I've talked to a lot of philosophers who are materialists, but not really, or dualists, or not really, and idealists, but not really. But these are nonetheless sort of categories that it falls into philosophically. But none of these kind of philosophical notions about the body, although they claim to be ontological, really address the question of what bodies themselves are. So that's the question that I really want to talk about. And I want to think about this from a really biological point of view. So I've put up on the screen a bunch of biological examples. And in fact, as we'll see later in the talk, all of these bodies are very closely related. But the conclusions we come to about our own bodies, a good test of those conclusions is, how well do they apply to bodies of other creatures, whether they're paramecia, like the guy up there on the upper left, or trees, or jellyfish, or whatever. So the biological answer, the basic biological answer to the question, what are our bodies are organized bunches of cells. And here are a couple of examples. The lower picture here is a picture of a pond in Yellowstone. And it's filled with a very complex community of bacteria. And you can see that although these bacteria are each individually microscopic organisms, their colonies are quite macroscopic. And these colonies are very highly organized. They're many species in here living together. Each of those species is doing a particular thing within the colony. So this microbial body is, in a way, not unlike our bodies. It's a similar size. It's not as complex, but it's very complex. There's a lot of communication going on in there, a lot of cooperation between cells, and also a lot of competition between cells. This upper picture, which looks like kind of an abstract art form, is in fact a picture of connections between neurons in the human brain. And they're color-coded for different types of connections, but it gives you an idea of the complexity of the relationships between cells. And keep in mind that in the brain, all the connections between the cells are implemented by the growth of the cells themselves. So you have neurons in your brain that are, easily about this long because they have to get from, this part over here to this part over here, or they have to get from this part in here to this part way back here in the parietal cortex. And if you look at your body as a whole, then you have neurons that go from your spinal cord down to your foot. So, these guys are individual cells that are this big. So communication in the body is implemented by cells with these very different shapes. There's a lot of organization. So when we say organized cells, the emphasis is really on the organization of the cells. And biology is a science of organization. And that's really what I want to bore into a little bit today and think about our bodies as organizational entities. And of course, in the last 20 years or so, we've realized that human bodies aren't made of just human cells. In fact, our bodies are about 10% human and about 90% microbial. And this just shows some of the different types of microbes that are found in different parts of our bodies. It's useful to remember that your body is basically a hollow tube, and it's got a channel through it that starts in your mouth and ends in your anus, and most of the bacteria live somewhere in there. And your body's full of little sort of pooched in areas like your earlobes. Your lungs are the biggest one that are also full of bacteria. So we're complex ecosystems of many, many hundreds of different kinds of bacteria as well as human cells. And this is true for every other complex organism. So insects, trees, all complex organisms are highly populated by microbial symbionts of one kind or other. But I'm going to focus on the traditional view of bodies, which is this one, which is that the body, at least of a complex organism like an animal or a plant, is made up of it's a clone. It's made of the descendants of a particular cell, the zygote. And what I'm showing you here is, this is the worm, Caenorhabditis, which is a very well-studied organism. In fact, it's the only animal for which we know in every individual where each particular cell came from in terms of how it derived from the zygote. And this picture is a summary of the first few layers of those cell divisions. But this traditional view is that an organism's body is a population of cells that are all descended from a single cell, the zygote, and they organize themselves in various ways. And during this developmental process, some cells die because that's what they're supposed to do. That's how they fulfill their communicative function in the development of the organism. And this extends to all kinds of organisms. So this is the development of the frog embryo from one cell to hundreds of thousands of cells. And in the early stages of this, you can pick out any one of these cells and isolate it, and it will grow into a perfectly good frog. But at some point in the process, the cells start to take on individual identities, and then that's no longer true. So if you interrupt development early, everything's fine. If you interrupt development late, then the cells already know what they're going to do, and you don't get a perfect individual from just one cell. Now, this idea prevailed for a long time, and it's only really been modified by inserting all of those microbial symbionts that we talked about earlier. So you can think of your body as this clone of human cells surrounded by bacteria. Okay, so let's look at this process now very explicitly in space-time. And it looks like this. We start out there on the left with the zygote, which is one cell. And I'm separating out, in the picture, the red stuff is DNA, the genome. And then the black lines represent the cell membrane. And so the first thing the zygote does is divide in two. And when we look at that in the lab in a movie, what we see is one cell, and then it sort of pulls itself apart, and then you have two cells. But when you look at it in space-time, what you have is one cell and then it branches. So the important thing to bear in mind here is when it branches, the DNA divides, but it's the same DNA. One strand of the DNA stays the same in each daughter cell. And the membrane stretches out, but that membrane is continuous from the mother into the daughters. And the cytoplasm, the stuff inside the membrane, is similarly continuous from the zygote into the daughter cells. And that continuity is preserved at every division that follows thereafter. So if you look in space-time, this is all one entity, right? It looks to us, looking at snapshots in time, like a bunch of different entities that are glommed together. But looking at it in space-time, you can see the continuity. And you can see the continuity at the level of the genome, at the level of the cytoplasm, and at the level of the membrane. So each of us is very literally one object in space-time going back to the zygote. So when you talk about the persistence of the body, viewed in space-time, you have very literally one entity that's growing in space as it moves through time, but there are no discontinuities. No cell is ever destroyed, and no cell is ever created from scratch in your body. So your body really looks like this when you stretch the time coordinate out. Each organism is one continuous entity. So where did this zygote come from? And again, you see continuity. I mean, your zygote, the zygote that was you at one point, came from a large part of mom and a little bit of dad. And in particular, males are defined as the sex that just contributes DNA to the zygote. And the female contributes everything else. So when you hear about epigenetics, it's almost all a mother effect story. There are a few paternal effects, but the cytoplasmic memory all comes from mom. So the memory that your cytoplasm carries is your mother's memory. And every cell carries that memory. The memory that your genome carries is a combination of mom and dad. And that's just the way the male-female split works in mammals. So this bottleneck of taking multiple cells and squeezing them down into one cell happens for each of us. It's what creates the zygote out of this other highly differentiated entity with lots of parts. So this is one organism. Now let's expand the scale. And this is a picture that some of you have seen before at SAND in San Jose. It's from a 2015 review article on phylogeny, and it illustrates how organisms are related based on the most essential genes in their DNA, the genes that build the proteins that do things like DNA replication. And what it shows is that most of the organisms that exist, most of the biological diversity that's out there is microbial. And all of the plants are up there where it says plantae in letters so small you can't read them. And all of the animals are up there under metazoa. Everything else you see is single-celled. And everything other than this part look microbial. They're either bacteria or what are called archaea. And the archaea are organisms that look just like bacteria, but biochemically they're much more like us than they are like bacteria. And they were only discovered about 40 years ago. Before that, we had no clue about early evolution. And in just a year, after a lot more DNA sequencing, we get a much more expanded picture. This was from Nature last year, and it's the results of some sequencing experiments going out in the oceans and in hot springs and places like that and just isolating random microbes and sequencing them. And this picture tells us something that's extremely important, that was explicit in the last picture, but it's really evident here. And if you look at that big sort of spray of colors at the top, those are all bacteria. These creatures down here are archaea, and the ones in green are the ones that are closely related to us. But what this picture tells us is that virtually all of the molecular diversity in life is in the microbial world. And microbes all look the same. There are very few distinctions when you look at microbes under the microscope. And you say, what does this thing look like? Well, most of them they look like little spheres, or they look like little rods, or they look like little filaments, or something like that. But that's about it. All of the morphological diversity, the stuff that we say is interesting about bodies, arms and legs and brains and hair and livers, leaves on trees, all of that is concentrated in animals and plants. And those are these little blips down here. So this tells us that DNA diversity, this huge thing that this represents, and morphological diversity, which is what we're interested in with respect to bodies, are actually anti-correlated. So most of the DNA diversity is located in a part of life that has almost no morphological diversity. And all of the morphological diversity is located in parts of life that have almost no DNA diversity. Now that says something important. It says that the memory that makes us look the way we are, the memory that defines our bodies, is not genetic. Because there's very little diversity between us and mice. There's not much diversity between us and that C. elegans worm I showed you. We have about the same number of genes in our DNA as fruit flies do. The genetic diversity is all over there in the microbial world. The memory that makes us different is in the cytoplasm and the membrane. It's the memory we get from mom. It's not genetic. So epigenetics is a really big deal because epigenetics is about what's going on in that non-genomic memory, and that's most of it. So that's just sort of a side issue. What I really want to focus on is the last common ancestor to all of this diversity, which biologists call LUCA, and point out that that was a cell. So we had a cell a long time ago that was our last common ancestor of us and trees and microbes and everything else, and at some point it divided. And those divisions were just like the division of your zygote. They preserved the membrane, they preserved the cytoplasm, and where the DNA came into the picture is a question, theoretically. It may have come in late. It's not known whether LUCA had any DNA or not, or whether LUCA's genome was entirely RNA. But we do know that LUCA was a cell and it divided. And then we know that at some point down the line, parts of the bacterial lineage got together with parts of the archaeal lineage and created, by fusing, the cells that look like our cells, eukaryotic cells. So let's take this picture again outwards in time, and we get this. We get an ability to look at all of phylogeny as the developmental biology of LUCA. LUCA looks just like your zygote, and all of life looks just like your body. So all of life is descended from one cell by cell divisions that preserve memory, that preserve the membrane, preserve the cytoplasm, preserve the genome. So our bodies are actually continuous in time, not only with each other, but with all other animals, trees, paramecia, bacteria, etc. We're all part of this one growing entity that in space-time looks just like our bodies. So what we think of as our individual bodies are, in a very deep sense, not individual at all. They're little pieces of this vast growing structure, all of which arose from one cell. So all of which looks like the progeny of a single entity. So Luca is basically a zygote. And we can ask, where did it come from? What was the analog of mom and dad getting together? And where it comes from is a picture that looks structurally a lot like the previous picture. It's pretty clear that if Luca had DNA, then the bits that went to form Luca were RNA, not DNA, because RNA can self-replicate and DNA can't. So in the beginning, to form the first cells, three things had to get together. We needed RNA, we needed salt water, and we needed membranes. Membranes are the thing that hold it all together, that make it a cell, that make the fundamental division between me and the outside world. And there are a lot of different ideas about how this actually happened and what actually went on in this purple circle that's labeled combinatorial chemistry, which just means chemistry that happens by lots of stuff getting together. And I know lots of you follow Bruce Damer's work, which was just published, and he's going to be at SAND in the fall, I think. Is that right? Sorry. So this is a picture from his 2015 paper, just illustrating his particular theory about how this combinatorial chemistry worked. And he basically combines ideas from several previous models in a way that introduces the notion that the way to get the combinatorial chemistry to work is to cycle over and over and over and over again between wet environments and dry environments, and hence between hot environments and cool environments. And in fact, that kind of cycling is how all combinatorial chemistry systems work. Systems meant to build different kinds of drugs, systems meant to synthesize DNA, etc. So it's a picture that, from a chemical point of view, is very understandable. And it gives a suggestion for where these parts came from. And remember, these parts are basically the components of your memory. The RNA is what gets converted to DNA in advanced cells. That's your genomic memory. The salt water is what becomes the cytoplasm of your cells that serves as the medium in which everything happens. And it's very highly organized. It's not just goop. So that's part of your memory. You actually, every one of your cells remembers that environment in which all of this took place. And the membrane components are actually the membranes around all of your cells. They're continuous through time with those membranes. So where does that leave us? It leaves us with this picture where organic molecules in salt water, subjected to a combinatorial chemistry process, gave rise to a cell, and we are the progeny of that cell. And we are continuous in time with that cell on multiple levels. We have memory structures that not only preserve a snapshot of that cell, but are actually continuous with that cell. There are no breaks. There's nothing sort of created de novo. And we are parts of this body, but so are all other living things. So our bodies, in a very important sense, are not our own, and they're not even bodies. They're part of someone else's bodies, right? They're part of Luca's body. And Luca's about 4 billion years old. So we are each parts of a 4 billion year old body. So what are our bodies? They're kind of like those little bits of water. They're parts of this moving developmental process of an enormous organism that fills the entire planet and that's very, very old. So if that's what our bodies are, what are we? And I'm just going to leave you with that question because I think it's a fascinating question. What are we, given that our traditional view of what our bodies are is actually fairly grossly wrong? So I'll take questions, and I think we have at least 5 minutes left.
To what degree do you think, focus on the understanding of this biological view of the development of life is at odds with or in support of the non-dual view of the universe versus the materialistic view? You know, i.e., consciousness being primary versus matter being primary. Because I intuitively, at least in my framework, which could be lacking, I feel like this is more and nothing wrong with that, but just wanted your clarity on that. Like this is kind of more in support of the materialistic view of the world, and time and space is, versus the non-dual.
Okay, from an ontological point of view, clearly this is a story about molecules and creatures. But I think a value of this kind of thinking from the point of view of theoretical biology or systems biology is that it does start to break down some of the barriers between organisms and between species and even between whole blocks of the phylogenetic tree. It, in a sense, forces us to see life not in terms of lots of individuals, but in terms of an individual. And I think from a more philosophical point of view, we have this concept of evolutionary biology and a concept of phylogeny that rides on top of that, that's very much formulated in terms of competition. So the Darwinian worldview is survival under selective pressure. However, if you look at the sort of underlying philosophy of developmental biology, it's much more centered on cooperation. So one thinks of the cells of the early embryo cooperating in order to develop more complex organs, for example. So if you think of evolution as a developmental process, it forces you to think of evolution as a process that's largely cooperative. And it forces you to think of evolution as a process of information exchange between different kinds of entities that are parts of the same body, in the same way that one thinks about embryogenesis as a process of information exchange between different entities that are parts of the same body. So the competitive view of evolution doesn't go away, right? There's even evolution, there's even competition in embryology, but it's moderated by this view of cooperation. So I think that's another philosophically useful lesson in this context of thinking about individual versus other, and what do boundaries mean in terms of interaction between a system and its environment. And I guess I see from my perspective, much of the question about what duality or non-duality means is what is the actual function of the interfaces that interactions cross? What does it mean to talk about interaction? Okay, Eric says, time is up.
Thank you,
Thank
Chris.
you very much.
Yay.